Thank you, Stephanie. Thank you so much, and welcome everybody who is here on the line with us this morning. I want to welcome you to the Pediatric Retinal Research Foundation's Family Connection Conference 2022. It's hosted by the Family Advisory Committee. My name is Louisa Rekia, the Community Outreach Coordinator, and I'm happy you're able to join us today for the conference. This event was made possible by the very generous donors of the Pediatric Retinal Research Foundation. So I'd like to share my appreciation to each and every one of you who joined us today for your continued support to our mission. Please drop in the comments where throughout the world you're joining us from today. So everyone on the call, please go ahead and share that with us in the chat. As you'll see on your screen, I think it's the next one. Yeah, as you'll see on the screen, today's agenda includes a research update, Q&A from questions that were submitted with your registration, an engaging breakout room session, a presentation from a very special guest speaker, a robust live Q&A session, followed with a brief closing message. We hope each portion of today's event gives you valuable updates, information, and excites you for the future that lies ahead. But before we get started, I'd like to mention a few reminders. This meeting is being recorded, and the link will be emailed to you within the next week. As you've probably noticed, your microphone was muted. Please remain on mute until the breakout session. Feel free to submit questions in the chat feature at the top or bottom of the screen. We will do our best to answer as many questions as possible, but please know we won't be able to get to all of them. So a little disclaimer. Also, keep in mind that the information shared at this event is for general informational purposes only. It does not constitute the practice of medicine or the delivery of medical healthcare services, including the giving of medical advice. I'll now give everyone a moment to read the slide for more details. Wow, hello from New York, hello from Chicago, the UK, love this. Michigan, Ontario, Canada, love it, love it. Germany, welcome. New Orleans, Ohio. All right. Now, without further ado, I'm honored to introduce Dr. Tony Capone. Dr. Capone is the PRRF president, as well as an internationally recognized clinician surgeon and educator. He has co-authored over 300 publications in peer-reviewed medical journals, book chapters, and publications from clinical trials. Dr. Capone is currently a professor of biomedical sciences at Oakland University and professor at the European School for Advanced Studies in Ophthalmology in Switzerland. He's recognized in particular as a surgical innovator. And he also serves as co-president of the ARC, which many of you know is the leading academically oriented private retina practice in the world. Dr. Capone completed his undergrad and medical degrees at Brown University, his residency at University of Pittsburgh, and fellowship at Emory University. Dr. Capone, it is my pleasure to turn it over to you.
Thank you, Louisa, for the very kind introduction, and welcome all. We're delighted to have you all here at this inaugural Family Connection Conference. Welcome to people from across the world, as well as to our friends from a number of other pediatric retinal disease message boards and societies and associations, including the Norrie's Disease Foundation. It's great to have you all here, and I hope you end up having a sort of rich experience in the next couple of hours as we share some thoughts and experiences related to the small universe of conditions that fall within or under the umbrella of the foundation. We'll be hosting the live Q&A session shortly, so please go ahead, and if you've not already submitted questions, so we have a number of them, as Elise has alluded to, go ahead and send them in, and we'll try to make sure we cover that material. It's my privilege to next introduce you to Dr. Kimberly Drenzer. Dr. Drenzer is my partner at Associated Retina Consultants. She's one of the team of 3 pediatric retina surgeons. Kim's an MD PhD, and her PhD work is in molecular genetics. She is really the anchor and the brains behind our pediatric retinal research laboratory. And so it's really a pleasure to turn it off to Kim, who will give us a little bit of a research update. Kim?
Okay, let me unmute myself. Well, I'd like to say good morning to everybody. It's such a wonderful format for everybody to get together and talk. So as Dr. Capone mentioned, that you know, I am a clinician, but I also have a real passion for research. And one of the things that I really loved about the Pediatric Retinal Research Foundation is that it is one of the few foundations that really links translational research. So we have the opportunity and the tools to be able to see patients and their unmet needs, and where biotech is not promoting innovation in new therapies and management, and we can bridge that gap, which is pretty exciting. I think I'm going to probably review research from a backward standpoint. So I, several years ago, over a decade ago, became very, very interested in retinopathy of prematurity and familial exudative vitreoretinopathy, in part because of the similarities between the 2 diseases and how they overlapped one another. However, one was due to being premature and the other was due to a presumed hereditary change in one of the genes in one of the pathways. So the current state of research is that we are very, very close in preclinical development to a therapeutic which will likely be beneficial to a large majority of patients with familial exudative vitreoretinopathy. That coming forward, like I said, we're in the preclinical phases, in the manufacturing phases. And we recently, just this week, received FDA approval as an unmet need for an orphan disease, and that the therapy would actually, per their review, looks very promising. So that's a huge, huge accomplishment, not just to me, but to the whole team working on that. And it's obviously the research project that we've been able to get closest to clinical trials. So what we've done with that, like I said, I'm going to work backwards. So what we have is we have a therapeutic agent that looks like it will be effective for a large number of patients with familial exudative vitreoretinopathy. And it may actually be able to expand after clinical trials beyond that. How we got to that has a lot to do with the foundation and its support. When I started this project, very little was really understood about why people had fever, why there was asymmetry between not just eyes, but between family members carrying mutations. And we weren't really able to make any connection to it until we had a robust biobank. And so essentially what an ophthalmic biobank is it's being able to say, I don't know what we need to do and look at genetically at this point, but I'd like to have that genetic sample available so as we get more information, we can do the appropriate studies. And it's a very powerful way to address unmet needs, because unlike typical research projects where I have to have a specific target and why I'm collecting that sample, this is a cumulative database, so we can keep expanding. We're only limited by resources, both financial resources and staffing resources. How many samples can we bank? How many can we prepare? And how many can we analyze? But that goes back to back in 2003, I started the biobank, recognizing that we had this unbelievable patient base that was getting great care from a surgical and all the tools we had at that time, but we weren't completely able to address the underlying issue, and that was very frustrating. So the biobank took actually quite, probably 2-3 years before we had collected not just enough of the genetic samples, but analyzed them and started to put together what I like to call the genotype phenotype characteristics, so we could make some meaningful understanding of what pathway was involved, why it's involved, how it plays a role in retinal development and retinal health maintenance. And it's one of the reasons that we had the big drive about the sequencing. So as I think most people probably are aware, medical technology changes very quickly, which is really exciting, but it's almost sometimes hard to keep up with. And the technology that we started back in 2003 was really very outdated and not high throughput. And it's constantly becoming more effective and more cost effective, both in time and money. We can do more samples, we can look at more genes for less money, which makes the biobank more robust. And that was really the impetus of the drive to get the new MySeq, which was next-gen sequencing. And there are even subsequent software technologies that we can use to address and use kind of the template that we used for addressing fever and being able to come up with a very, very exciting potential therapeutic agent to meaningfully change the way we manage fever. We can actually take that same template from the biobank and use that to address other rare eye conditions, such as, I've talked with a number of parents about retinoschisis. There are some exciting gene therapies in that area. There's gene editing, but you can't do any of this without having a robust biobank. And one of the rate limiting steps for most foundations is volume. If you just don't have enough patients with these relatively rare conditions, you can't target them. You can't come up with meaningful translational research. And so I think that one of the things that I think is great about the Pediatric Retinal Research Foundation is it targets a lot of unmet needs, not just from a research standpoint, but from patients and parents and portals and links and outreach. But the research is also very, very unique, and we have a huge ability to address all of these rare diseases because we have the volumes in the biobank. So I just wanted to take a few minutes to kind of explain, like in a backwards fashion, why the biobank, which is somewhat, I think, hard to understand why that's so robust and important, but it allows us to really do the important translational research, which goes into potential therapies. And as not just the technology from sequencing and genetic evaluation is emerging and improving and evolving, we have the same thing with genetic interventions. I'll speak a little bit to gene therapies and gene editing, which we are currently not doing, but I certainly monitor these where different states of research are, because I am very interested in, as I said, stepwise platform translational research. X-linked retinoschisis is a perfect disease, eye disease for gene therapy. The presentation and characteristic eye findings correlate very, very highly with a genetic alteration in the retinoschisin gene, about 96%. So if you look like you have retinoschisis, 96% of the time they can identify an alteration or a mutation in that gene. So that is the very, very first check mark to gene therapy being successful, is not having, part of the problems with retinitis pigmentosa is that there are over 100 genes identified, and each gene has over 100 mutations identified with it. So you're talking about an exponential number of gene therapies to try to target a disease. Retinoschisis doesn't have that problem, so they're a perfect gene therapy candidate. The current clinical trials have been very disappointing, mostly in the sense of the way that the protocol was designed. But more recently, I was reviewing a technology which is still in preclinical phases, but is a more robust viral vector, and actually delivers, the protocol is to deliver the viral vector where it needs to be, which is actually adjacent to the retina. So either a subretinal injection or an intraretinal injection for retinoschisis. So that company has some very exciting proprietary technology, but that's clearly in their pipeline and they're in the preclinical phases. The other thing that comes up a lot and is very exciting is gene editing, and the name that comes up with that is CRISPR technology. So the difference between gene therapy is that traditional gene therapy is taking the full gene and inserting it into the host cell, and that works very well for diseases that don't have what's called haploinsufficiency. It means that a normal, healthy gene will override the pathology caused by an altered gene. So the first step of traditional gene therapy is that you need to have not too many genes affected, and you need to not have haploinsufficiency. You need to be able to prove that a healthy gene will kind of override or overcome the altered gene state. Retinoschisin actually does check both of those boxes. So I do think it's just a matter of time before there is a viable gene therapy option for retinoschisin. Other inherited diseases that are a little bit more complex, multigenic, different genes involved, they maybe work in different ways, are potentially going to be more amenable to CRISPR technology. So CRISPR technology is gene editing. So when you gene edit, what CRISPR technology is doing is it's actually taking a genetic scissor and you're targeting the altered gene sequence, or the mutated. Generally, it's a single amino acid or the most amenable to this, and you're cutting that out and you're splicing in a different genetic component. So it has the benefit of being very individualized and very kind of designer technology. So you can take lots of different mutations. That's also a little bit of the rate limiting step, and it's in its infancy. So I think it gets a lot of hype, and I think it's great technology, but I still think that we're probably a little ways out from seeing that really come to fruition as a mainstream genetic therapy. But for sure, it's going to play a role in genetic treatments in the near future. When I say near future, I'm talking probably clinical trials, probably within maybe starting in 2 to 5 years, really coming to fruition as a common household name for genetic alteration, probably closer to 5 to 10 years. So that's kind of on the genetic front. There are obviously a number of different rare eye diseases which require a lot of investigation and a lot of research, and some are more amenable than others. But that's pretty much, I don't want to go into too much tangent discussions, but that I think really kind of explains the goal of PRRF, how we kind of interweave the clinical management, the clinical need, identifying unmet needs, and really using, for people who've kind of been curious about how the MySeq plays into this in the biobank, this is really the foundation for being able to make meaningful translational research that we can then do pilot studies in the lab to see if we have something that may be of therapeutic value. And I think I should probably wind up there.
Can you say a little bit about the growth of the biobank and how long it's been operational?
Sure. So the biobank, I actually started back when I first joined ARC as a fellow in 2003. So we have the largest biobank of rare retinal diseases in the world. The second largest is out of Leeds in the UK, which is pretty robust, but it has probably maybe half of ours, and it's actually much more targeted. So whereas our biobank, I set it up so that we could take it for, I wanted it to expand to all comers. The one in Leeds is a little bit more targeted, specializing in fever. And the issue with it, and it's kind of a funny thing, is that, Mike Tracy would tell, when I first came, would tell a very, very funny story at that time. Paul Sieving, who was going to be the National Eye Institute director, and was for several years, I think about a decade, maybe even longer, originally was at University of Michigan. And we are traditionally a surgical practice. And so a lot of our tertiary referrals are surgical. So we see very, very severe disease. Medical retina specialists tend to see much less severe disease. And so Mike would tell this very funny story of how he would sometimes have families with retinoschisis that some would be seeing Paul and some would be seeing Mike, and it had to do with the severity of disease. Were they relatively mild and stable, they would see Paul. If they needed surgery, they became Mike's patient. And we had the same issue that I'm very good friends with the person who runs the biobank out of Leeds, Carmela Toombs. And so she and I have been at the same symposiums and same meetings many times, and we will take that time to grab a coffee and compare what we're seeing in our biobanks. And she's referred medical retina, and we're mostly referred surgical retina. So our biobanks also look very different, even just for the entity of fever. And that, I think, is actually very, very telling as to how you develop therapies. So one of the most important things is that the ability to have a high volume concentration of patients with rare eye diseases in one practice was a huge opportunity. And that's why I really wanted to be able to harness that back in 2003. And so one of the questions also that comes up a lot about the biobank, and I think it's really important to understand, is that if we get useful genetic information that's helpful for families, for family planning and understanding, we certainly share that with the families if that's what they want, but it's not a clinical diagnostic lab. The real goal of the biobank is to promote research in areas that nobody else is looking at. And I'll just tell a slightly anecdotal story, which is for vision and ophthalmology, ARVO, in either 2005 or 2006. I had finally had enough volume and analysis of our biobank, and I did a poster, and it was the only presentation on Norrie's disease and on the Norrie disease protein, which is the gene NDP. It was the only one in a huge research and clinical forum to share information. And it was really completely a brushed under the rug gene. The whole pathway, everything, the understanding was very, very poor. The next year, there were 10. The following year, there were over 100 presentations at ARVO. And so that's, something that is very powerful for gaining understanding, is being able to bring to the forefront and bring to the public eye in the research world an area that is poorly understood and really not researched because of lack of volume. And I think that's one of the greatest strengths of the biobank in the sequencing program that we have. And I will just say that we got a very, very nice grant, $150,000 grant through the combination of Oakland and the Pediatric Retinal Research Lab. And one of our co-researchers, Dr. Ken Minton, we got a Carl's Foundation, I think it was $150,000 to support continued sequencing and analysis of the biobank. And now that's a huge support to being able to, because aside from just accumulating the data, so we accumulate the data or we accumulate the samples prospectively. Basically what we do is we take a blood or a sputum sample, we extract that down to DNA. DNA frozen at -70 is stable for possibly forever. And then that allows us to have this repository of DNA samples that we can then subsequently set up gene panels and investigational pilot studies through the sequencing, which is, like I said, it's becoming more and more cost effective. It is still expensive and requires staffing. And so that's it. That foundational grant was a huge, huge ability for us to maintain not just the sequencing we're doing, but expand our sequencing panels to other rare diseases as well.
I mean, a couple of overarching themes, but thank you, Kim, for that kind of detailed review of what's going on in the lab and a little bit of history. So, with broad strokes, the biobank is something that is unique to us with regard to the, again, small universe of diseases that we tend to. It's almost 20 years old now. Next year, it'll be 20 years old. And focused on the diseases of interest to everybody on this call. The sequencing that we do has been to a meaningful degree facilitated, made possible from donations from to the foundation, helping us to purchase a sequencer and supplies to do that kind of work. So sort of the support of the community has been critical in that regard. And I think Dr. Drenzer nicely sort of spoke to the fact that by virtue of the work that we do on this fairly focused population, been able to identify a factor that's important for vascular development, and now in the midst of working on developing a drug used to treat vascular issues, primarily in the kids. And in that pathway, we will talk a little bit in the Q&A about the path from the lab to the shelf in the pharmacy, if you will, figuratively, where you can pull the drug off the shelf and use it. But the orphan drug designation or recognition by the FDA is a really key piece, because it greases the skids. Just to kind of summarize briefly the advantage of that, it greases the skids with regard to accelerating development, et cetera, if it's recognized to address an orphan disease. So all of these things really have sprung out of the lab, and frankly, none of them would be possible without the support of the community. So really appreciate the summary on that, Kim. And we'll be chatting about some of the other diseases of interest in a bit more detail, also in the Q&A that's kind of come through in the chat. So we'll be hitting a lot of things in the time that we have.
Sure. Yeah, I was going to mention too, in case folks have submitted questions and it seems like we're not answering them or ignoring them, I just want to reassure you, we do have a full Q&A that we've pre-recorded that we'll share at the end of the conference. We've addressed a lot of these questions in that Q&A. So if you feel like we're purposely skipping a few things that you're asking, it's a good chance it's covered in the later Q&A. So that's why you may not see us answer some of those coming through. So actually, I'm going to read a couple that have come through over email and things like that as well. And then Dr. Capone, if you want to field some of the ones you've seen in chat, you can address some of those as well. But let me start with a couple that came in. There was a number of questions of people that have asked about juvenile X-linked retinoschisis, wanting to know about clinical trials, research updates, any information or new things that you can share about that particular form of retinoschisis?
Kim, do you want to catch?
Yep, I can take that. I was going to say, yep. So there There were two clinical trials that were recruiting for a gene therapy, which was gene replacement therapy. Both used a recombinant adeno-associated viral vector, which is very, very common for eyes for a lot of reasons. And retinoschisis is a relatively small gene, so it's very amenable to an AAV vector. One of the companies, I believe AGTC, left that trial due to recruitment issues. I think through the NIH, they are still actively recruiting and trying to make some modifications to their protocols. One of the biggest issues with the protocol, in my opinion, is two. One is that they were really trying to do intravitreal injection, and AAV is not a great vector for intravitreal injection. It's not the most robust virus. It has a hard time getting to the retina and infecting the host cells through a vitreal approach. And there were some reasons why people wanted to do that, and they're reasonable. You can make it an office-based procedure if you can do it as an intravitreal injection. As I mentioned earlier, the biggest problem with AAV is, for the most part, you really need to put it where you need it to infect cells. So you need to put it on the retina, in the retina, or under the retina. So subretinal or intraretinal injections give you the best chance for success. So I think that the active clinical trials have really struggled, not because the therapy isn't valuable, but because the protocol design was not ideal. And I mentioned that there is another company using a proprietary AAV vector. And what they've done is they've altered that vector to be more robust in its infectivity, essentially, so that you can potentially infect more cells. But they also do recognize that it needs to be their proposed or right now, what they're thinking is going to be their protocol is a subretinal injection, which is more realistic. Because of what they call a lateral spread, they don't think they're going to have to put it in as potentially dangerous sites from a subretinal injection. They may be able to put it further away from the central macular region, and it may spread infective-wise into the more central vision areas. What remains to be seen is whether or not this is going to, give you sufficient robust expression of the retinoschisin gene with a single injection, or if you're going to need multiple injections. 6 months down the road, you may need another one. And they're still in preclinical phases, so they have not started clinical trials. But like I said, I think that I feel that it's inevitable that a gene replacement therapy will be available for retinoschisis patients. It's not going to start off being available for all patients, and this goes to any clinical trial. The way clinical trials run is that you first have to show safety. So your phase 1 studies are, safety is the measure. Efficacy is not the measure. So those are usually people with very, very severe disease with, for lack of a better way of putting it, not a lot to lose if it's not safe. In rare diseases, you have the benefit that you don't have to do nearly as many patients as you do in non-rare diseases. There's an expedited pathway. Phase 2 is where you continue your safety trials, but now you're starting to look for efficacy. By phase 3, that's when you've proven safety, you have an efficacy signal, and now you're doing a more robust trial to really show that you have an effective drug. One of the benefits that, and it sounds a little bit weird, but if you get the FDA in particular, if they identify not only that you are treating a rare disease with an unmet need, but they also deem that your therapy looks promising, because there's no other intervention, they're somewhat lenient on the efficacy side. So they will allow, the most important thing is proving safety, and they will allow a fairly rapid expansion to patients that maybe aren't showing efficacy, because what they want to do is roll out that agent as quickly as possible. So their number one goal is showing safety. Efficacy, they almost look at it as a real world situation. We want everybody to use this because it's safe. And if it works, we want everybody who's a potential candidate to get it. So it will seem very, very limited early in the phases, but unlike, let's just take diabetic retinopathy or macular degeneration, something that's very, very, common in the adult population, that will have to go through a lot more rigor before they kind of open up and expand the profile. So it seems very limited early on, and you'll feel like you aren't, a lot of patients with rare diseases are not going to meet the criteria in the early phases, but it will expand much more rapidly in a short term. So that's kind of how rare diseases work in that setting.
So, obviously, if you have rare diseases, you're not going to have the opportunity to test the drug in 1,000 patients. So the expedited pathways kind of takes the rarity of the disease into consideration and tries to get you sort of a practical path from the lab to the clinic. But as I'm listening to Dr. Drenzer's explanation, I think maybe a couple of big picture comments would be in order here. So, and we'll take the example of X-linked retinoschisis since that's where we started. So the main treatments right now are gene therapies by delivering the correct genetic material through a viral vector. And for those of you who are not in science, you can basically take the shell of the virus and take out the DNA or the RNA and use that essentially as a microscopic syringe. So you take that virus, you remove all of the virus DNA or RNA, depending on the virus, and then you put in the stuff that you want to inject, and then deliver that to the site, and then the correct genetic material using the viral coat, in essence, as a syringe, as I mentioned, injects the correct genetic material into the cells. And then at a certain percentage of the time, that genetic material will be incorporated into the cell and start to produce the correct protein. It isn't at 100%. So there are a bunch of sort of steps that are critical with regard to how efficient they are. And that's what Dr. Drenzer was talking about with regard to this, the particular vector that is being used is this adeno-associated virus. And you'll hear that a lot or see that a lot. If you look into the genetic literature, it's AAV. And AAV has a number of problems, as Kim mentioned. Especially early on, it engenders, some of the studies demonstrated engenders a fair amount of inflammation. Secondly, the infection rate, meaning the number of times that the virus will attach to a cell and inject material, is relatively low. And then remember that once the material is injected, the proportion of times that material will be incorporated is also low. So now you've got a double low here. Also, we don't talk about this a whole bunch, but the viruses vary with regard to what we call payload. And that is kind of what you would imagine it would be intuitively. It's how big the virus is and how much stuff you can get into it. And that also limits what conditions you can treat, right? The bigger the genetic replacement that you want to do, obviously, the bigger payload you need. So all of these things factor with regard to what diseases you can treat, how efficiently and effectively you can treat them. And then the last point I would make is Dr. Drenzer was mentioning, intravitreal injection. And again, for those of you who are not in our world, intravitreal injection is the kind of injection that we do in the office now for many diseases, where you come in through the white of the eye and you just inject the material into the vitreous gel that fills the center of the eye. And then you expect the material is going to diffuse and lay out on the surface of the retina, and some of it will be delivered there. So that's another sort of barrier to efficiency, right? Because now that material is going to have to diffuse in the whole vitreous cavity. Only some of it will get to the surface of the retina. Only some of it will be effectively injected. Only some of it will be effectively incorporated. So you get a little bit of an idea by that last sentence as to some of the challenges to genetic therapy. And then again, Dr. Drenzer mentioned some of the approaches that are being tried. There is a lot of genetic therapy going on right now. I think that's the upside. We are going to kind of crack this nut, sooner or later. Some deliver it directly beneath the retina as well. in an attempt to bring it right to the point of interest. So that's one thing. That's the kind of the science of the genetic injections for non-scientists. The other thing is that for all the retinoschisis studies to date, they're not interested in treating infants and children. They're primarily looking for young adults or adults, frankly, for a variety of reasons. We can go into the details of that, but a theme that carries through to treating the constellation of diseases that we are interested in here is that these diseases have varying degrees of severity many times. The same gene may present in a parent in a mild fashion and in a child in a severe fashion. And that's often how we end up incidentally finding it in the parent, right? The child's affected severely, the child gets brought in, we make the diagnosis, we go, this is often hereditary. We look at a parent, we often find more subtle evidence of disease. So that's a concept known as expressivity or degree of expression. Like the same gene in different people can manifest itself in varying degrees of severity. In any event, to fully correct some of these things, when the children are born, they can already have moderately advanced disease. So the approach is going to be different to address those children. And there's a question here about ICSI, sort of the technology of fixing early stage embryos genetically and things like that. And we'll talk about that a little bit, and it's part of the recorded Q&A. We touch on that there. So Kim, is everything I said correct?
Yeah, perfect.
Tony, this is Mike. I just want to add something. Paul Sieving and I did the categorization of the classification of retinoschisis many years ago. And for a period of time, I've met with the study group from NIH relative to X-linked retinoschisis. And Kim or Tony, I can't remember which of you mentioned this earlier, that there's basically medical retina and surgical retina. And the NIH has got wonderful medical retina people, still do, and very little in the way of surgical retina people. And so consequently, the intravitreal injection, as opposed to a more proper AAV type injection into or under the retina, wasn't performed because they really didn't have the appropriate manpower to do it. That's continued at UC Davis, where Paul is now located. And I'm still getting phone calls from doctors about the technique of that intraretinal or subretinal injection. And so this newer technology may be helpful for that. But the reason basically was they were just uncomfortable with that type of delivery system. And it became very obvious when you met with them that they really wanted an intravitreal injection just because they were much more comfortable doing that.
Mm-hmm.
Thanks, Mike. That's great. So I'm going to hit on one other. There's a question about X-linked retinoschisis question, and then we'll transition to some of the other stuff. But the buccal vitrectomy question regarding X-linked retinoschisis patients. So sometimes patients with schisis will develop retinal detachments. That usually requires that they have that happens when they're older, not invariably, but usually. And buckles are only used for that instance where they develop a retinal detachment in addition to their retinoschisis. When they're treated, appropriately with a scleral buckle and usually a vitrectomy at the same time, in other words, fixing the eye from both the outside and going inside, the success rate is actually very respectable, very good, probably in the 80 to 90% range. Regular retinal detachment in an adult is in the 90-95% success rate with a single procedure. So given that these are considerably more challenging technically, 80 to 90% is pretty good. Usually best, of course, in experienced hands because they have some, unique features, and like everything else, the more you know about, if all you do is fix Ferraris, you're a great Ferrari mechanic, right? So schisis is our, excellent schisis is one of our Ferraris, but it's just that we see a lot of it. And go ahead.
I would just add to that real quickly, because I think this might come up. I have a reasonable number of patients with X-linked retinoschisis that had a combined, both they have a schisis cavity, a fairly significant schisis cavity, and then developed a rhegmatogenous component, retinal detachment. And sometimes they don't live in Michigan and they're being, co-managed from afar. One of the things that comes up is that somebody who's not used to dealing or seeing that will confuse the schisis as a either recurrent detachment or an incomplete repair. And I think that just bears some mentioning, because I think it causes, understandably, a lot of concern and anxiety. Somebody goes back home and hears a differing explanation as to what's going on with the eye. And so just when you fix it, the success rate is very, very high for fixing the detachment. It doesn't mean, since we don't have currently a genetic intervention to put a functioning retinoschisin protein back into the area, the schisis still persists. So we sometimes can make it better with surgery, but it's very, very common that the detachment portion is fixed. And that's what the true success rate is, that when you're fixing a retinal detachment. It doesn't mean that we've eliminated the schisis portion.
No, that's a great point, Kim. So I'm gonna pull on that string a little bit more. So when an adult has a retinal detachment, the goal and how we define success is complete repair of that attachment. The retina totally attached again. In pediatric retina, complete reattachment is often not possible, number one. And number two, there are a number of other abnormalities, unlike the adult where the retina developed normally and detached. Some of the children that we tend to have retinas that have not developed normally. So when we fix them, open quote, close quote, that's not analogous to the adult, where we basically bring the retina back pretty close to its normal state. So we're drifting a little bit onto the clinical side of things, which is fine. But so for folks, particularly folks who are far away, we usually develop a partnership with someone, local. And because it's important to have boots on the ground. And part of that relationship is a conversation between us and the local or referring doctor about what we're shooting for here. I.e., so if I get in a child, the retina 50% was totally detached and I get it 50% attached, I'll send them home saying, things look great, we're 50% reattached. And they see the local doctor and he or she says, oh my, it's 50% detached. So we need to have that glass half full, glass half empty conversation with the folks back home. And that's a big part of long distance care for these rare diseases. Kim, there's a question here about T-span 12 relating to fever and the distinction between X-linked fever and non-X-linked fever. You're the best person for both of those.
All right, I'm going to start with the easier one first, which is T-span 12. So T-span 12 is a really, really interesting gene in the way that it relates to eye development and eye maintenance, and in particular, retinal development and retinal maintenance. T-span 12 is, the best way to think of it, is a chaperone for the protein neuron. So T-span 12 helps direct neuron binding and neuron pathway activation. T-span 12, however, is actually expressed ubiquitously. It's expressed in lots of cells, lots of tissues. It's functioning in a wide variety of manners in Wnt signaling all over our body. And the reason I'm just is talking about this is because when we're talking about X-linked fever versus Norrie disease, we're talking T-span 12 and its role in neuron, and then we're going to talk about neuron. So they kind of overlap. T-span 12 generally has more mild disease than a lot of the other affected genes. And part of the reason is it's twofold. One is because TSPAN12 in the eye augments the action of neurin. So TSPAN12 plus neurin is 2 times more effective than neurin alone. But if you don't have TSPAN12, neurin still functions. It just doesn't function quite as effectively. So it's a suboptimal signaling. So we tend to see less severe disease. When we see more severe disease with TSPAN12, it has a lot more to do with retinal folds than, and maybe a tractional element, but not a full detachment or bleeding or lipid, a lot of the exudative. Part of the reason I think is that aside from the fact that neurin can function without TSPAN12, is that I also think that very, very severe mutations in TSPAN12, because it's utilized in so many different signaling pathways throughout our body, probably has more lethality. I think for an infant to make it to term and to be born and be relatively healthy, they actually can't have very, very severe mutations in TSPAN12 because of all the other systems that would be affected by it. So it's not to say, and people, misinterpret this, it's not to say that you can't have poor vision with TSPAN12. It just means that the severity of the pathology is less than we see in some of the other genes, such as, Frizzled-4 mutations or LRP5 mutations, which tend to have more problems with, chronic and relentless exudation.
Can I do a quick sidebar here? So to explain a couple of concepts to folks. So first of all, genes vary with regard to how important they are, to development, right? So not all gene mutations are equally severe. And the second thing is not all mutations, even we get within a given gene, are equivalent. And that's because your genes encode for proteins. So if you get a mutation that allows pretty close to full function of the protein, you're going to have either almost undetectable disease, or mild disease. Conversely, that same gene, if it's severely crippled so that the protein is minimally or almost non-functional, that's going to be severe disease. So you can have a spectrum of impact, even if you're affecting the same exact gene, depending on how much of the gene is, how much of the protein can be produced and how functional that protein is. So that explains the variability that you'll see of different mutations, of the same gene. I'm sorry, Kim, go ahead.
So that was great. That's very helpful because it also segues to, now we'll go kind of 180 degrees. So neurin functions better with TSPAN12.
Kim, can I interrupt for 1 second?
Sure.
I often feel that the Wall Street Journal is the most current medical information. But next to that, the patent office comes in terms of new information. And you might just mention about TSPAN12 and need to have neurin with it. That's been argued in the patent office for some time, actually.
I don't think there's a huge argument because the patent itself clearly states that it needs to have exogenous neurin to be functional. So there's what Mike's referring to is that there's a TSPAN12, basically it's a TSPAN12 mimic antibody developed by Genentech. And in their patent, what they found was that they actually need to add neurin in order for their therapy to be effective, which is what I was going to segue into, which is saying that, so TSPAN12 needs neurin, and neurin doesn't need TSPAN12, which now goes into—
That's the point I was trying to make.
Yeah.
Yes, exactly, yeah.
Which now segues into Norrie's disease versus X-linked fever. And this can be very, very frustrating. I think that neurin is one of the most interesting proteins, maybe ever, just in the sense that it's very unique in the sense that it's really only highly expressed in 3 tissues. So it's in the retina, it's in the cochlea, in the inner ear, and it's in our brain. And it really doesn't play a major role in other tissue systems. It may play some minor roles, but not a major role. What also makes it unique is that it's very rare. From an evolutionary standpoint, our genetics and our bodies have developed to have what I would call backup systems. So when you have one gene that doesn't function, there's a sister gene that can come in and at least take over the function. If it's not 100%, it's at least effective enough to not create a problem. So Norrin's unique in the sense that there is no backup gene, there's no backup protein that can take the place of Norrin. And because Norrin is really only highly utilized in these 3 tissues, it's not lethal. So you can have a healthy full-term baby because it doesn't affect the other systems. But it's absolutely required for the development of your retina, your cochlea, and a good portion of your central nervous tissue in your brain. So when you have either no Norrin or you have a mutation that essentially renders it unable to fold properly, so it looks like you don't have Norrin, you have the most severe presentation, which is the classical description of Norrie's disease. And that's why the triad is blindness, hearing loss, and deafness, and cognitive decline, because there's no backup protein to save the lack of Norrin. Where it gets very confusing is that X-linked fever is also a change, a mutation or an alteration in the Norrin gene, but it's not functioning as if you don't have it. So the Norrin is able to fold enough and have enough of its appropriate structure that it can activate the pathway somewhat. And this has been done in lots of labs, and we've looked at it, which is the severity. So you can have a big spectrum, as Dr. Capone was talking about, depending on what the mutation affects and how much it knocks out its function determines kind of the severity of the disease. So X-linked fever and Norrie disease is really a spectrum of the NDP gene being affected. And the severity depends on really how much you're knocking out the function. From, you'll have some mild Norrin mutations, which maybe only knock out 20% of its effectiveness. You can have pretty decent retinas with, pretty reasonable vision and not real severe disease. Those patients actually, they have found now that they tend to have sensory neural hearing loss at a younger than anticipated age, not severe enough that it really stands out, but they will notice that it's more than just an age-related change, that diminution starts a little bit earlier on in life. You don't really see, the brain does have one other gene, it's Wnt7, which can also help to back up Norrin. So you don't see with a mild Norrin mutation, much, if any, cognitive change. And then there's a spectrum of that. So that gets to the point that Norrin doesn't need T-span 12, but T-span 12 needs Norrin. And so that's why there's kind of these overlapping presentations and severity based on these mutations, which makes it very, very complicated. But that's one of the things that without this biobank, you wouldn't have ever been able to really parse out these details.
Great, Pam, that was great, great answer. Louisa, Stephanie, do we have anything else?
Yeah, sure. I'm going to switch to the clinical direction a little bit here for a couple questions, and then we're going to wrap up with the live Q&A so we can save time for the recorded one.
Okay.
A couple of the really great questions came in. One is, there's a whole team of specialists that are supporting your child who has vision loss. The retinologist, the ophthalmologist, the optometrist, vision therapists, pediatricians, parents, family members, teachers. This person is hoping to better understand the roles and relationships that each play with one another to help kind of keep track of everything as a parent, because it can be a little overwhelming. So how can you guys best describe how to approach working with this team of individuals to give the best care to your child?
That's a great question. So, Kim, if you don't mind, I'll start and then
Mhm.
you can weigh in. As well as Mike, you have the most experience with this, but I think the short answer is it takes a village. There's no doubt about it. And in some regards, I share with parents many times, we have the easiest job. Technically the work is meticulous. It takes a long time to get good at it, et cetera, et cetera. But our interaction is for a short period of time to sort of optimize the anatomy as best we can. But then really the success that, one will have after that depends on, the, the degree to which, the retina is developed and the degree to which, we get it reattached. And those are factors that, you as parents can't impact, but the biggest thing to do after we kind of put things back together as best as they can be is the follow-up care, particularly with regard to things like patching and monitoring for glaucoma, glasses, and all that kind of stuff. So the aftercare, honestly, and respectfully, I think that's where all the heavy lifting goes. But all the parents, on this, feed, I'm sure are nodding, because once you leave, from the surgery, the weight of all of the rest of that falls on parents. And
Mhm.
from a medical perspective, the point person typically is the pediatric ophthalmologist, because they are the person who are kind of command central. They will weave in whoever else you need, whether it's for a pair of glasses, for a glaucoma evaluation, finding you a low vision specialist, and things of that nature. So from a medical perspective, we're kind of a special teams player. And once we do our bit, we have our seat back on the bench, and the pediatric ophthalmologist becomes, the quarterback. But at the end of the day, and I know I recognize many of the parents on the call, the ultimate responsibility for the captaining the team accrues to the parents, and it's kind of like whether you like it or not, because no one is going to care about your child to the degree that you do, and so you end up being the primary person who's charged with making sure that all the T's are crossed and the I's are dotted to the best degree that you can. So it's a job. It's a big job, and our part is important. But in some regards, I'm not being overly humble here. I think our part is the easier part, because it's a time limited thing, and the heavy lift really accrues to you as parents and the other caretakers. Kim, Mike.
Well, I have these children that have more severe disease often require multiple operations. And so I think, even though your description, I think, is correct in terms of the pediatric ophthalmologist directing things prior to that, but I think that it's important to have the child examined by the surgeon, who may pick up things that the pediatric ophthalmologist might not be in touch with, or the other people in touch with. So I think it's important for the surgeon to continue to follow the child at some time frame that they feel is important relative to possible reoperation.
Yeah, I would just add also, I think that it's a really frustrating thing, because you're trying to weave in different specialties, and just in general, I think part of the, what I would just call, rehabilitation, therapy. So you see this with people who've had strokes, you see with people who've had, hip surgeries. Some areas are better than others. And I think that visual rehabilitation can be very, very frustrating, because there's no clear pathway. There's no clear entity that you say, and here's the group that will, do a great job with this. It's a little bit of a gray area. And I know that that's where a lot of frustration comes in, because, you know, for a lot of patients and kids, they're never going to have a quote unquote absolutely normal looking retina. And so I think it's very frustrating to have somebody who maybe isn't familiar with the surgery, isn't familiar with the condition, and looks in and says, well, there's nothing I can do because that retina is not normal. It's not going to function. And that's not true. And we know that. So you know, it can probably feel like an uphill battle, I think, a lot, to be always advocating for, well, there is some function and I want to optimize it. And I know that the retina is not completely normal, but let's use what we have, because that's really what we're doing. And I think that it's not always appreciated in any sort of visual rehabilitation, unless you're really dealing with somebody with a lot of experience, which there just aren't a lot, that children's brains are very flexible at rewiring. So if you took an adult and you know, structurally totally altered their retina, they would not rehabilitate well. But a child is very, very different, because the plasticity in their connections in their brain are so much more flexible. And I will, you know, so I did, when I was an intern, I did a couple months on neurosurgery, and I'll never forget, there was a 6-year-old boy with a cerebellum mass, a huge tumor in his cerebellum. And I was, you know, scrubbed in, doing the surgery, and essentially he had no cerebellum left. And we took the cerebellum out, and I remember saying, like, well, how is he going to, so your cerebellum is important for your basic functions. It's what you need to breathe. It's what you need to have any sort of stability walking. And, you know, you take out a cerebellum in an adult, they might not even live, let alone do well. I saw this little boy 3 months later. He was walking, he was running, and they would do all these PET scans, and they just showed that he just rewired all those functions to a different part of his brain. And so that, I think, is a really important thing to understand. And you just, as frustrating as it is, you just kind of have to be pressing as an advocate, saying that, I know it's not looking like the prettiest retina, but let's make what's working work, because that brain is still active and able to make those connections. So that's kind of and I'm very frustrated with it as well. I mean, I really do wish that we had a better pathway towards what I would call visual rehabilitation.
Yeah, I would make one comment along just to close this off before we go to the recorded Q&A. A lot of times, folks who haven't got experience in this area will look at the retina and see that the anatomy is, you know, clearly not normal, and jump to conclusions about what the child is capable of based on sort of the degree of abnormality of the anatomy and what they would conjecture would be the case if it were an adult. And it's often mistaken for the reason that Kim just mentioned, that the children are incredibly plastic, and they do, what that means in English is their brains are able to do a lot more. They will squeeze every little bit of vision and function out of whatever it is that they have in a fashion that all of us who are adults, if we lost what we have down to the level that, you know, they have, it would be a huge and dramatic loss, and we haven't got the plasticity to adapt to it. So all the time, you know, kids come in with retinas that you wouldn't imagine would see very much. It's one of my favorite games to play with, you know, the fellows and residents that we train is, you know, the child's asleep on the table, we're examining, and I'll ask, guess what this child can see? And almost always people will underestimate based on their conjecture from adult work as to what a retina is capable of. So that's a great point, Kim, and that's, you know, speaks to the relationship building with the local doctor, the part of what we do when we talk to them, because it ends up impacting on patching. So we'll get a retina attached, the other eye is normal, and they'll commonly say, well, this abnormal eye is not really capable of very much, so why bother? And the answer is you'll get more out of it actually if you treat it fairly aggressively. Even if it's not normal, you'll get the best that it's capable of. And the way we think about things is a child born today has a projected lifespan of 100 years. Everything we can do to optimize, you know, their vision really means more fertile soil for tomorrow's technology to fall on. So, and if you don't do that for the child, you're making a decision for them for 100 years. So that kind of drives the full court press kind of rationale with regard to both optimizing anatomy and optimizing vision, even in an anatomically compromised eye.
I have just a really quick comment, Tony.
Sure, Mike.
It may not be everyone aware that I'm a couple of years older than Tony and Kim. And I've come to a period now where I'm receiving letters from patients who often have these retinas that the doctor locally was going to give up on. And they're thanking me for 25, 30 years of vision. So it can be that this type of visual function lasts a reasonably long time. Maybe not 100, but 25, 30 is pretty good.
We're delighted. Welcome back, everybody. And hope so far that you've enjoyed the information and opportunities for discussion today. We're looking forward to your feedback as we will take your input as to into how things worked out today in terms of what we craft in the future. It's particularly nice to have an opportunity to get together with folks who are not like-minded and like circumstanced. I'm sure, as everybody knows here, that there aren't a lot of folks in your neighborhood who have similar issues, and it's good to be able to chat with folks who understand and have experience in all of this, from the medical care side to the day-to-day living side. I'm very excited to introduce to you today's guest speaker, Brendan Werner. At 6 months of age, Brendan was referred to Dr. Tracy, whom we saw earlier, shortly after he was diagnosed with fever. And Brendan's mom, Alyssa, was a fierce advocate for her son, which is, I think, a strong theme that runs through kind of the parental part of our organization. At a young age, she encouraged him to master Braille and focus on excelling in school and getting actively involved with sports for the visually impaired. These included baseball, goalball, and rock climbing. After high school, Brandon continued his studies at Albion College, working several part-time jobs as well throughout his undergraduate experience. Following graduation, he found he was unable to secure employment, and that is a recurrent theme for some of our kids who are able to make it through the educational structure and then have their challenges entering the workforce. Not to be defeated, he returned to school to earn a master's in vision rehab therapy from Western Michigan University, and then success followed as he was offered a position at U of M, University of Michigan, currently working as an accessibility analyst focused on making websites and software easier to use and accessible for everyone at the university. It is now my distinct pleasure and privilege to introduce you to Brendan.
Can everybody hear me all right? Quick sound check.
We can hear you just great.
Okay, great. Thank you so much for the introduction. I guess I'll start out quickly by just describing my job. I work as an accessibility analyst at the University of Michigan, and my job is to primarily analyze digital assets. I would say basically how I describe it. So everything the university has, from like websites, software, different textbook delivery platforms. We look at those things that different departments have at the university, and we sort of provided advice to those departments on how to make those resources accessible so that folks who attend the college with different disabilities are able to access them. So I guess the first slide we have up here are the main themes of my presentation. The first theme that will be recurrent through this presentation is self-advocacy. Basically, everybody needs to communicate their needs, but if you're someone with a disability, that's especially so as you're going through school, college, and then once you get your job, what is it that you need to be successful? I need to communicate that to others. I think it's really important. Think carefully about life choices. This one will become more evident as I give examples throughout the presentation, but really it's hard to know what you want to do when you're young, but thinking about the things you want to do and whether they're possible with your blindness, I think is very important. It's one thing I think is important because I went to school for economics. I wasn't able to get a job with that degree. I had to, I went back, took another path of vision rehab therapy and was able to get a job in that field. So thinking very carefully about what you want to do, I think is very important. This third theme, creative use of resources to accomplish tasks, applies to services, technology, and other things you may have access to, tools as you progress. Okay, go ahead and switch the slide. So this slide focuses on extracurricular activities primarily. I called it things to think about when you're in school. I participated in band and choir. I think that having an activity, it really doesn't matter when, but when you can be successful at is very important as you are progressing through your younger years and into high school and into college. It allows you to prove to others that you can do things, because in the activity, you will most likely encounter folks who say, oh, I'm sure you won't be able to do that because you can't see. When I was in band, folks were very hesitant about my ability to actually march on the field. This is very important to learn how to prove people wrong, because when you go into college and into your job, you're going to run into the same thing. People who think you can't do these things because of your disability. Also, socialization, it's these extracurricular activities give you a way to socialize, which is extremely important. They can lead to networking opportunities. People who I went to school with and were in activities with, I actually, some of them happened to be working for or attending the University of Michigan, and I was able to be introduced to their friend circles, introduce them to mine. So I think this is really important, especially with a disability, to make sure that you start that base and can socialize. Okay, go ahead and switch the slide. So I really think that two of the most important skills for folks who are blind are mobility and mastery of technology. Being able to find the door of where you want to interview and where you need to work is really important. Navigating around, obviously this is more challenging when you can't see. And if you have good mobility skills, I think it really gives you the opportunity to prove to people in the job setting, in the school, that you're confident. As you walk around, your demonstration of confidence, like this is people's first impression of you seeing you. These next things I want people to think about is, especially in regards to classes, this is going back to the thinking carefully thing. Think carefully as you take classes on whether, it's something you can do. Blind people can definitely do, and we know that they're successful in degrees like law, music, stenography. I'm in technology. But again, I don't want to dissuade anyone from any career, but I have friends that have taken chemistry classes, and in the labs they had someone sighted sit next to them and help them through all the labs. So you really have to think, if you're the hiring manager, are you going to hire someone for a job that requires an assistant, right? I mean, this is really important. I mean, if you need an assistant in the chemistry class, again, I don't want to dissuade anyone from doing anything, but you have to think really carefully about the field you choose and whether it's going to be possible to do this field with a disability, because what you don't want to do is go to school for 4 years and then be stuck not being able to get a job. Okay, go ahead and switch the slide. So this slide is kind of things to think about when you graduate high school. I tried to gear it for folks who are on a college path, or those who might decide that they don't want to go to college, just get a job directly after high school. So making connections with professors is extremely important in colleagues, because you never know when these sorts of resources might be helpful. If you need your mail read, if you need someone to read you something quickly out of a book, if you're friends with the person behind the desk at your apartment or your dorm, the person in the cafeteria, any one person is going to get probably irritated if you ask them too much for help. But if you ask many people and you keep track of who you ask when, then you can ensure you don't ask the same person for help too often, and you don't sort of strain those connections. One thing I do, that this probably sounds crazy, but I'm a technologist. I often would try to use like an Excel spreadsheet to keep track of who I've asked to read me things how often, and then I can make sure that I'm doing things in there and for them in return to try and keep it equal. Again, maybe this isn't for everyone, but it's something you can think about. Picking the correct tool for the job. This is really important, especially for totally blind folks who are using screen readers. I gave an example here of iOS and the computer, but iOS VoiceOver on the iPhone is a great screen reader. It really has changed my life, and I think it probably has for other blind folks as well. You can do so many things with it. You can read your mail, make phone calls, read letters, detect currency. But one of the things it's not good at is creating formatted Office documents, because it doesn't tell you about indentation. So, and really, we're living in a visual world. If you don't format a document properly, if it doesn't have bullets and numbering and things align properly, it reflects badly on you. And this is actually something that in college, people let slip for me, which I really wish they hadn't. And then really my first boss at University of Michigan sort of really showed me how important this was to do the formatting. Again, the screen reader on the PC is much better at this. So knowing when to use each tool. Again, I'm happy to answer questions about tools if you contact me after, but I just want to give this one example because I think it's one that is a common one that folks can relate to. GPS was a game changer for me. I started with the Trekker Breeze. I still use it a lot. You can get GPS programs for phones, although I think that there's still a place for the dedicated GPSs like the Breeze, and I think there's a new one called the Trek now. They're really good because in winter climates like Michigan, you can use them with gloved hands in a coat pocket. Touchscreens don't tend to work with gloves. I know there's some gloves that makes them work, but it's really nice to have a dedicated hardware device, and GPS has really gotten me out of a lot of jams. And I know my other blind friends who use them, it's really, really been a helpful thing as well. These GPSs also allow you to mark places like, entrances to buildings that might be in the middle of parking lots. If you're totally blind, parking lots are really tough to navigate. Bus schedules and ride sharing. Obviously, if you can't see ride sharing like Uber and Lyft, this is like a game changer. I mean, both apps are accessible. I think, talking to people about bus schedules is a particular problem. I found for me being totally blind, they're oftentimes in inaccessible PDF files. So again, this is about the self-advocacy, doing research where you need to go, ask about bus routes. You can call the transit center and ask, those friends, those colleagues. Just the other day, I needed to create a account, and it was one of those things where you had to type the characters in the picture. And I had a friend who hadn't asked for help in a long time, so I gave him a call and asked them for help. These things are really good resources to use. Okay, go ahead and switch the slide. This next slide, I'm going to talk a little bit about job searching. This is so much easier than it used to be in the past. With websites allow people to search for jobs, whether they're low vision or blind. You can use a screen reader or you can magnify the website. Indeed.com has been really implementing web content accessibility guidelines to make their site accessible. And you type the information in once and you can apply to multiple jobs. I mean, this video conferencing interviews is a game changer. One of the hardest things for me when trying to get a job was getting transportation to all the interviews. But with COVID, it's actually, in some ways, for a blind person, I think it might be easier to get a job right now because so many interviews take place online. So, for example, I have a blind friend named Kyle who just got a job in New York. And what he does is essentially makes jobs accessible for other blind folks. So for example, if someone is trying to get a job in a call center and the software doesn't work, then he'll make the software work with the screen reader or the magnifier so that they can do that job. And he lives in Michigan, and he was able to receive that interview over Zoom and then once he got the job, he flew to New York, and he really would have had no way to get to New York. I mean, the funds to go on that interview, especially when he was, interviewing at so many jobs all over the country. Okay, next slide. Okay, so LinkedIn, again, is really important today. Good news, Microsoft bought it, and Microsoft is really passionate about accessibility, and they're improving the accessibility of LinkedIn. It literally gets better. Every week I see accessibility improvements in LinkedIn. So some job duties may be more difficult for you to complete, so you have to really build and really excel at those you can do well. Some parts of my job, I asked for assistance. There's some parts of my job that are visual, but other parts of my job, like, editing documents and writing and communicating, I can do those things well. So I pick up the slack on some of those areas and have my colleagues help me with some of the things that are more difficult due to not being able to see. So always thinking about things you can do to make yourself more marketable. You never know when you may lose a job. And then, obviously as someone with a disability, it's going to be harder to find employment again. Certifications and classes. I mean, with remote learning now, it's so easy again to go online and take a class. If you want to go somewhere to take a class to meet people, you can rely on Lyft or Uber. I mean, these things weren't around when I was in college, which was really only like 5 years ago. So this just shows how quick these things are changing. I can take a Lyft down to the community college where I live, where that used to be a 30-minute bus ride. Now it's a 10-minute Lyft, and it's $15 to get there and back. So it's really no more expensive than buying gas in a car. One thing I'll mention too about these ride sharing services, they have programs you can sign up for that give you discounts, like a 15% discount on all the rides. I did the math, and for me, the amount that I use these things, I come out hugely on top to be able to save 15 to 20% on all the rides. So then this kind of goes back to the tools thing and resources. So repeatedly trying tools is really important. If a tool doesn't work for you, you should continue trying it once in a while. So as I guess give this an example. The Mac has a built-in screen reader called VoiceOver, and it just doesn't work with the tools I need to use at work. I use Slack to communicate with other teammates at my work. I use Google Docs, and VoiceOver just doesn't work with those. But every time, Apple releases a new operating system every year, I download it and try it and see if it's improved. And that really goes with anything, I think. And also the tools that, I guess that what I would say is the tools that are best for one person aren't necessarily the best for the other person. I gave the example above of VoiceOver. It's not the VoiceOver on the Mac. Again, I use VoiceOver on the iPhone a lot, but VoiceOver on the Mac isn't the best tool for me. But if you were someone who is doing a lot of writing, VoiceOver does work very well within Apple's iWork suite. So like Pages and Numbers and Keynote. So well, in fact, that if someone was a writer, I might say that, that would be the way to go. And I've actually recommended that to people again, because again, if, for me, I have to use Google Docs, but, if you don't have to, people at my work collaborate with Google Docs, but if you don't need to use that, you use other tools, then, it might be another piece of technology that's best. So don't, I guess let people say, oh, this is definitely the best technology. Do the research yourself and try everything and find out what works best. So that's the end of my presentation. I guess, I don't know the timing, but if anyone has questions, I'm happy to answer a few questions. Also, I don't have the chat open. So if there's chats, if someone could read them, I turned off the chat so that I could focus on listening to my notes for my presentation. So I'm not receiving chats with my screen reader as well. So if you have a question, either speak it up, or if someone wants to chat, then maybe someone else could read it.
That was great, Brandon. There were a lot of great tips there for everybody. I really liked your Excel spreadsheet to keep track of, who it is that Joe favors to. I think that's pretty I would probably say let's open it to see if there are any questions from the community.
Yeah, yes. So please go ahead and use the chat. And just so everyone knows, Brandon did share with me that anyone who would like to reach out to him after today, he is sharing his email and contact information. So please, any questions, comments, or anything you'd like to reach out to Brandon for, he did give permission to do that. So Brandon, thank you very much.
One more quick thing I'd like to share too. I would say making friends, like, becoming good friends with your professors and teachers, I think is a really good strategy. There is a certain thing that they're required to provide by law, but if you're actually friends with them, they're going to do more. As an example, in my undergraduate degree, I got to the point where, I was kind of that weird kid that like went to visit the professors, like, hung out with them on the weekends. But, I coded with them and had lunch with them, and in exchange, they did a lot for me. They would take their notes off the board and type them in a format that I could read with my screen reader. I mean, one professor, we did visual electronics, and he literally set up a breadboard, with, which is like a, big circuit board. He actually showed me the components, like this is a, transistor, this is a resistor, and we hooked things up. I mean, and other, other professors have read books to me. And, you can be creative, like think of favors you can do for them too. Maybe, I don't know what they might be, but maybe, helping them with chores or something, and maybe they'll, help you. It's really, I would say that this building relationships with the professors, I know I mentioned it in the presentation, but it's really important if you want to succeed. Because then there's, like I said, there's what they can provide by law, but then there's what more they're going to provide if they like you, if that makes sense.
Honestly, Brandon, I have 3 kids in college, and I think everything you said is good advice for any kid in college, really. The professor engagement piece is big. No better way to show that you're interested.
I mean, even like, professors would take the time to, take a hot glue gun and hand draw graphs in economics class with hand, hot glue gun and construction paper so that I can feel them. I mean, they don't have to do that. That's not really their job. And so, if they like you, then, they're more willing to do that.
So we do have kind of one interesting question, I think, out of the chat is what video games, or if any, would you recommend for folks that are visually impaired?
So there's the 2 of my favorites. There are so many, there's a website called audiogames.net where you can learn about them. Gosh, I don't even know where to start with this. There's so many. 2 of the games I like are older. One of them is a racing game called Top Speed, and it's Top Speed. I think it's up to version 3. It's really interesting because you can actually play online with other people. It's an audio driving game that says left, and you wear headphones and you can hear where you are. That is an incredible, to be able to play with your friends online is really cool. For the social aspect. Another one I'd recommend, it's abandonware now, but you can, if you Google 3D Velocity, it's a fighter jet simulator for folks who are blind. It's really good. I would say it's as good as a sighted game. There's a good story, a great story mode to play by yourself where you have to fly over this island of someone who's like, it's like this evil, it's a typical thing, like evil, cloner wants to take over the world, and you have to stop him, and you sort of fly over his island and, like launch missiles into different key like operations that he's got going. And then there's also a good online mode where you can take off with all your friends, and you got, 6 or 7 planes in the sky, and you try to shoot each other out of the sky. So, that's, I mean, for the social aspect, though, that's really cool, those 2, because you can play with your friends. So, there's lots of them that are single player, but I try to pick the ones that you can play with other people. That's what I find to be the most rewarding is you get, a group of friends together on like, like sighted people do, they get on these, chat programs like TeamSpeak or I don't know, Mumble or something. And then they, they all play games together. And if you're blind, you can do the same thing.
Great answer. I wouldn't have guessed that there were any options, much less a website that's a repository for them. So that's awesome. Brandon, thank you so much for sharing your journey with our attendees and making yourself available to folks who are interested afterwards. That was great.
And like I said, please feel free to reach out to me. Probably phone is best, like texting me. I can respond to emails, but the quickest answer will come from a phone call probably. I get tons of emails. So if you text me or call me, you can skip past all the emails.
Awesome. Well, thank you, Brandon. We're going to move on now to the last part of the conference. Dr. Drenzer and I reviewed the questions submitted with your registrations and answered as many as possible in the video that's going to play up next. So enjoy, and I'll be back at the back end. So this is a great question for a couple of reasons. I think, first of all, it underscores the distinction between a genetic diagnosis and a clinical diagnosis, right? So you could diagnose fever by testing somebody genetically and seeing if they had one of the recognized genes which cause fever, or we can make the diagnosis clinically because it has a very characteristic appearance. So if we, see a child or patient and they have the classic manifestations, particularly if we then examine, the parents and one of them has the manifestations as well, it's pretty clear that they have the clinical diagnosis. So that's kind of the two different ways to do it. Interestingly, of those who have a clinical diagnosis of fever, when you test them genetically, only about half of them end up coming back positive for one of the known mutations of fever. So if folks are negative, it doesn't mean that they don't have fever. It just means that they don't have one of the genes that has been recognized. For example, it may be that it's not a single gene that causes fever in some patients. It may be the combined action of a couple of genes. And those genes that are acting together have not yet been identified, and things of that nature. So apart from that general comment, speaking about the special case of a child, there's a significant amount of overlap between fever and retinopathy of prematurity. And I would say that the diagnosis of fever in a premature infant generally comes about clinically, not genetically, clinically in one of two ways. Number one, if the child has retinal findings that in spite of the child being premature, these findings are atypical for retinopathy of prematurity and more typical of fever. So there are certain ways that fever can look with regard to the configurations of the detachment and so on and so forth that are more typical of fever than of ROP. And then the other way is a child who has retinopathy of prematurity that has an unusually aggressive course that doesn't respond to the conventional treatment approaches that typically result in a good outcome. So unusually aggressive retinopathy of prematurity is another thing that signals to us that a child may have a fever gene in addition to being premature, which is, throwing gasoline on a fire with regard to the rate of progression of the underlying retinopathy of prematurity type changes. Dr. Drenzer, anything that you would like to add?
I think that summarizes it very well. I think it's very, very important, especially when dealing with a premature infant, that if we have the suspicion of fever, the genetics are helpful when they're positive, but they're not determining the end result of the clinical diagnosis when they're negative. And really, it's helping us to better manage that child long term, because if we suspect that there's an atypical feature, which may be more in line with fever, it may prompt more lifelong examinations at a more frequent interval than we would otherwise also be doing for just a retinopathy of prematurity situation.
So we consider, fever is a lifelong disease, and we follow folks with fever typically at least twice a year, generally, for life. And the reason is that some of the features are present and even dramatically present in infancy. Other features are more smolderingly progressive. You know, they progress little by little over the course of time. And the three major features of fever in the back of the eye are traction on the retina due to scar tissue, leakage from abnormal blood vessels, and loss of the very smallest and finest blood vessels, which are known as capillaries. Much like in a plant where it's the fine hair-like roots that do all of the work. So in the body, the capillaries are really the point where oxygen is exchanged. And so in fever, the capillaries can gradually be pruned as an affected individual grows. And that can have consequences on vision, and that's potentially amenable, at least with current technologies, to treatment with laser and hopefully, you know, with other approaches in the future. So those are the reasons that we follow folks with fever lifelong. Scar tissue traction and the traction attachment that can occur as a consequence of it, leakage and capillary dropout. Kim, anything to add?
No, those are the salient features, and they're hard to predict. I mean, one of the things that we've learned with doing the genetics and trying to track some of the characteristics that we see with children is trying to be able to better predict so that we can maybe identify somebody who only needs to be seen twice a year as opposed to every 2 to 3 months. But essentially, we're looking for those characteristic changes to indicate how aggressively we have to intervene and how often we need to see the patient. So this is a very common question, both the COVID vaccine and its how it impacts eyes, but also just getting infection if you happen to become positive and ill with COVID. There's been no evidence or even theoretical indication that either contracting COVID or getting the COVID vaccination has any impact on the eye, and more specifically on the retina, even if the eye is at risk from something like fever or other retinopathies. There doesn't seem to be any overlap or concern in regards to following normal recommendations to keep the child safe.
Right. So I would just add, so therefore there's no reason out of concern for worsening fever to avoid your child getting a vaccine, or yourself getting a vaccine if you're an adolescent or an adult with fever. Yeah, so the challenge with, so retinal folds are one of the more common, unique fever features. There aren't a lot of diseases that cause the folds that have the appearance that the eyes do when these folds are present due to fever. And I think, the upside is that eyes with these folds tend to be very stable over the course of a child's life. So they do have a fairly significant impact on vision. But in spite of the sort of how dramatically these folds alter the anatomy, the children often end up seeing better than one might imagine, which is kind of a recurrent theme for sort of anatomic distortion in a child's eye. Children are often able to make more of the anatomy that they have than one might imagine. The downside is that we really haven't got away with current technologies to unfold them in a way that has an acceptable risk profile. I mean, so it wouldn't be accurate for me to say, well, I can't go in and unfold it. I can, or Dr. Drenzer can, but we have to cut the retina and do a variety of other things that actually make the situation are likely to make this, the treatment would be worse than the underlying disease, if you would. So likely stable over life, and with current technologies anyway, not really repairable.
So there are a number of therapies that are being looked at, and One of the ones that is very close to us is because we've been working on this for a number of years, which is a drug called Norogen. And it is a mimetic of human neuron. And what we found is that being able to add that into eyes with retinopathy similar to what we see with fever is able to rescue the retinal vasculature and provide regrowth of capillaries and also prevent the leakage that we see in these capillaries as well. And this has been done in animal models. We have not used it in humans yet, but that's probably, in my opinion, the most exciting therapy. There are a couple other groups working on therapies that are of a similar fashion and trying to work at ischemia. They aren't specific for fever. So this is the first one that's specifically looking at fever as the target eye disease to try to treat. In regards to where we are with the development of Norogen as a therapeutic for fever, we are currently in what are called the preclinical phases. So we are doing all of the manufacturing to get it up to the quality that it needs to be used in a human, and doing all of the supporting documentation, toxicity studies, animal studies. The hope is that we will be able to file with the FDA to move forward with clinical trials within the next couple of years.
Both aren't familiar with clinical trials, that Dr. Renzo is talking about entering phase 1 clinical trials. Clinical trials have 3 phases. Phase 1 is the safety phase. Phase 2 is when you look for what we call biological signal, basically to see if it works. And then typically in phase 3, you'll do a larger number of patients and check for both safety in a larger population and try to refine the specific dose. So if, for example, to play out the rest of the hand, if I may, Norogen were to make it into a phase 1 clinical trial within a year or 2, it's typically then another 3 to 5 years before it's available on the shelf, if you will, for the doctor to use it. So we're probably talking about 3 years would be very, very optimistic, more like on the order of 5 years or so before Norogen will be commercially available. Kim, do you agree?
Totally agree. I think that's a very realistic timeline.
So as regards the issue of financially supporting the research initiative, monies that are donated to the Pediatric Retina Research Foundation really go to our 2 major mission initiatives. One is to support the community of patients and their families who have the pediatric retinal diseases that are sort of in our universe, if you will. And the other is to support the research proper. So really the best way to support both the community and the research is to donate to the foundation.
Well, so as the goal with Norogen is to really address the underlying problem, the primary problem with fever, which is the lack of complete vascularization of the retina and the loss of capillaries. So it's really looking at the vascular issue. It has the added benefit of having a neuronal regenerative capability potentially because of the way that capillaries and neurons grow together. So that is on the forefront of something that would actively change the current status of the retina in making it healthier and less fever-like, for lack of a better term. Other treatments are still otherwise really geared more towards controlling the sequela of having incomplete vascularization and leaky vascularization. So controlling exudation, treating the avascular retina, surgically intervening when you have scar tissue or tractional changes. And those really continue to be the mainstay of treatments at this point.
I think that's a great answer, Kim. I think it's important, though, as a point of reference here, when we talk about treating fever, the paradigm isn't like, say, treating pneumonia, where you have someone who was normal before they had the pneumonia, then they had the infection, then you treat them, and they go back to being normal. That's not really where we are, where our opportunities are in the arc of the disease, if you will. When someone has fever, or for that matter, this is a conversation that applies to nori disease or Coast disease or some of the other things that we treat. Many of them have their onset in terms of when they start to impact the retina, in utero, during formation of the fetus. So when the child is born, we're sort of handed a certain sort of set of findings, if you will. And our charge as physicians is to try to normalize those features in as much as we can, whether it's medically or surgically. So rendering, and where I'm going with that is where we encounter questions about cure. So cure with currently available therapies and the current paradigms isn't really possible, because you'd have to have the opportunity to have, a normal substrate, or a normal sort of set of anatomic circumstances that you could bring the eye back to, like my pneumonia example. And we never get that with the kids who have the pediatric retinal conditions that we tend to. So we're relegated to try to, our current, sort of treatment initiatives are all geared towards trying to make things as normal as possible, given what we are handed when a child is born with one of these conditions.
So in my opinion, genetic testing plays a variety of roles. At this point, genetic testing is not going to be used for a cure. And so we'll start with that, because that tends to come up a lot, is how do we cure this? Fever is complex because multiple genes are involved. And in order to cure a genetic disorder, you really need to be able to replace the affected gene. And so when you have, the more and more genes you have that are involved, the harder it is to come up with a true genetic cure for it, because it becomes more and more complicated. So I don't necessarily think that genetic testing is essentially for a cure at this point, or for gene therapy, but I do think that genetic testing is very, very important for better understanding the variety that we see in how the eye behaves, how it behaves over time. So we do learn more about understanding, the different genes and what their role is in the health of the retina. A negative genetic test does not mean that you don't have fever, because it's a clinical diagnosis, but it does help support a fever diagnosis, particularly if there are other factors that may be confounding the diagnosis. And I also think that it's very, very important from a family planning standpoint. Oftentimes people are surprised that they have a fever mutation in the family. They can be sporadic, so it may just be that child or that patient was the only affected individual within the family, but that tends to be less common. So this will oftentimes prompt multiple family members to get tested. And then more than often than not, we find that we identify a number of patients in the family that didn't realize they have fever. They come in, they get their exams. Some of them actually have enough change in their retinas that they do warrant some sort of treatment or intervention. And furthermore, I think that it's important for future generations. As we do hope to be able to get better with interventions and treatments, and eventually maybe even gene therapies for cures, early diagnosis is going to be key, and maybe even in the early phases of pregnancy for family planning and medical planning. So I think that genetics is more than just saying, aha, we have this and we know how to treat it. And hopefully that's what it'll eventually lead to as well. But at this point, I think it still has a robust place in patient management.
Yeah, I can think of two other things, really. So if we're going to, so we examine family members, whether or not the affected individual, the child, test positive genetically. When we don't have a positive genetic test, though, we're relegated to the clinical examination as the only way that we can diagnose a family member, right? Whereas if we have a positive genetic hit, then even if someone lives far away, we can make a genetic diagnosis. We can make a diagnosis on genetics alone as to whether or not they're a carrier. But then to get back to the point that we started off the whole conversation with here, to assess the severity of the disease and whether or not they need treatment, to Dr. Drenser's point, will require a clinical exam. But to say whether or not they just carry the gene and therefore have the fever gene or one of the fever genes or several, genetic testing is useful in that regard. And then I think from a theoretical perspective going forward, as we develop therapies, there are some genes that we will be able to treat, first, second, third, fourth, and fifth, have to do with a variety of technical factors. And knowing which specific one you have, will allow us to identify what people would be sort of eligible for or amenable to the therapies as they become available. Hey, folks, we're going to end here, so we end on time and we're respectful of everybody's time. Hang on a second, I'm losing myself here. All right, there we go. So first of all, I hope that everybody found this an informative and enjoyable way to spend a Saturday morning. I certainly really enjoyed participating in this, and want to thank the whole group of individuals that made today's event possible. First, thanks to our donors who allowed us to offer this event to the community. It's thanks to your generosity that we can pursue our missions. I'd like to also thank the Family Advisory Committee members for the gift of their precious time and their input. Their thoughtful planning and feedback and efforts ensure that the event that we were able to host an event with relevant content. You'll find a list of all of the committee members in your workbook. I'd like to send a special shout out and thank you to Brendan for being our speaker today. Really enjoyed your talk. It's inspirational for all of us to hear about how you faced and overcame adversity from childhood through young adulthood, and just kind of the positive, practical perspective that you bring to describing those challenges and how you overcame them. Thank you, Brandon. That was really a great job. Lastly, I'd like to thank our breakout room facilitators for generously offering to give us their time and assisting in the initiative today. You'll all see their names listed on the agenda in your workbook. These volunteers have gone above and beyond to give everybody an opportunity to be heard and to connect with others, as well as to learn directly from each other in terms of real life experiences. Many of you have asked how you can get involved and support the work of the foundation. On your screen, you'll see 3 volunteer opportunities currently available. We've formed the Family Advisory Council made up of a select group of individuals and parents representing the diseases that are the focus of our foundation. This committee will help drive our strategic initiatives and ensure that the voices of families are represented in our programming and initiatives. We are still looking to fill a few more spots, and so please reach out to Louisa if you have an interest in learning more. We are eager to have you on board. We also welcome families to become ambassadors for the foundation, which means sharing your journey to provide inspiration and awareness for pediatric inspiration and awareness for engagement in helping to address pediatric retinal diseases. We are excited about hosting our 4th annual Hope Division Walk looking to the fall this year, and are currently seeing committed members interested in planning the event. So both the virtual and live components, as we've done in the last couple of years. To learn more about any of these opportunities, reach out to our team at info@pediatricrrf.org. And then to close, we'd like to invite everyone who's participated today to stay in touch with us. We have a number of great activities coming up and would love to see you soon. So for more info on the foundation and getting involved, visit the website at www.pediatricrrf.org. If you haven't done so already, sign up for our bi-monthly newsletter. There's a button at the bottom of the website where you can do so. And lastly, join us on Facebook and LinkedIn by following, liking, and engaging with our posts. You'll find our pages by searching at Pediatric RRF or the links on the site. So thank you again for your time, everybody. Have a great day.